So thank you. Hello, everybody, and thank you for joining us to another Mineral Life seminar. So today we have the great pleasure to have Dr. Jake Bailey with us, who is an associate professor at the Department of Earth and Environmental Science at the University of Minnesota. So the main driver of his research is to better understand the interaction between the biosphere and the geosphere, and in past and present setting. And one, among other topics, one of his main topics of interest is polyphosphate metabolism and its role in diverse environments. So that is today's subject. So we are really excited for this talk. So as usual, the whole seminar will last for 1 hour with some time for question at the end. So if you have a burning question, don't hesitate to write it in the chat and we will read it out loud at the end of the talk. So the floor is yours, and thank you again.
Great, thank you so much for having me here, and thanks to all of you for coming today. And today I'd like to talk a little bit about polyphosphate-accumulating bacteria and their role in influencing the formation of phosphate minerals and also potentially their dissolution. And we're going to go all the way from the kind of ocean environment all the way to the human oral environment. So I consider myself to be a geomicrobiologist, like many of you. You probably know what that is, but basically it refers to looking at the interactions of the biosphere and geosphere broadly, but more specifically looking at the role of microbes and their influence on minerals. And as many of you are aware, there are many known mechanisms for how microbes can influence either the precipitation or dissolution of minerals that could range from changes in redox, changes in pH, serving as kind of nucleation sites for mineral precipitation, and so on. Additionally, microbes in life, broadly speaking, can concentrate ions, leading to conditions that are supersaturated with respect to the mineral, which can lead to precipitation. Additionally, there microbes, that would be an example that would be here with the magnetosome formation and magnetotactic bacteria on the left. But also organisms could also deplete or cause a localized kind of depletion in ions, leading to undersaturated conditions, as is the kind of proposed mechanism here for boring in cyanobacteria that bore into carbonate rocks that are thought to kind of translocate calcium through the cells and create like a zone of localized undersaturation along the boring front, as proposed by Garcia-Pichel and co-authors. So today what I want to talk about is polyphosphate. And polyphosphate is kind of a polymer of phosphate groups that are linked by phosphoanhydride bonds. And it is kind of similar in some respects to ATP, and the energy is required for its synthesis, and the energy is released during its hydrolysis. So you can have basically a chain of phosphates from 3 to upwards of 1,000 groups long that basically makes a polymer that sometimes occurs as an inclusion body within the cell. And so polyphosphate is used by most, if not all, organisms at some level, at some stage in their kind of development or life cycle, often in kind of transitory or trace concentrations in the cell. And so it's kind of made and turned over relatively rapidly, similarly to ATP. But other organisms, not just bacteria, but other organisms, particularly bacteria, can accumulate lots of polyphosphate, up to 20% dry weight of the cell, oftentimes in inclusions. And so you can see here, these are cells stained with DAPI, and the kind of bluish color is the cell, and then the bright color here, the bright yellow, is basically the shift in the emission wavelength of DAPI when it's bound to polyphosphate specifically, that gives you this kind of yellowish color to the granule. And polyphosphate is used in a whole bunch of different ways by different organisms. It can be used as for energy storage, for phosphorylation. For example, it can phosphorylate glucose instead of ATP in certain select organisms. It can be used as for phosphate storage or for cation storage. It can be used for templating biominerals like bone. It can be used in stress response or to regulate transcription or to influence the formation and function of cell membranes and diverse other roles that it has been shown to play in cells. And polyphosphate inclusions can also, because there are these polyanions, can also commonly include concentration of cations as well, which could include calcium and magnesium, manganese, potassium, cobalt, a whole bunch of different things. Even things like nickel and lead have been shown to be concentrated within polyphosphate inclusions. And one of the systems where polyphosphate has been kind of extensively studied for a long time is in wastewater systems, where you basically have cycling conditions between anoxic and oxic conditions that it leads to the enrichment in polyphosphate accumulating bacteria, whereby you basically have under anoxic conditions, polyphosphate accumulating bacteria can use their polyphosphate stores as an energy source to take up carbon and store carbon as polyhydroxy alkanoates in the cell. And then basically they can use those PHAs under the oxic conditions to give them some kind of advantage metabolically. And then basically they can also take up and restore that polyphosphate under those aerobic conditions. So those are kind of a canonical system whereby polyphosphate is linked to these kind of cycling between oxic and anoxic conditions. But I want to talk about kind of two different stories here. And the first one involves polyphosphate in big bacteria. And the big bacteria that I'm going to be talking about are Thiomargarita namibiensis and related taxa. And Thiomargarita namibiensis is the sulfur pearl of Namibia, is what its name means. And these organisms are found in upwelling systems and also cold seep systems and so on. But particularly where we primarily study them now is off of Namibia, where you have upwelling waters that lead to high primary productivity on a shallow shelf system. And then basically when you have the degradation of that biomass, you have a high biological oxygen demand, which leads to conditions that are anoxic and sulfitic, both in the sediments and even into the water column. So you can have a shelf setting there that's variably and seasonably hypoxic and strongly sulfitic, so much so that there are sometimes eruptions of methane and sulfide into the water column that cause these big patches of water to be kind of like milky white in such a large area that they can be seen. They look kind of like a phytoplankton bloom on satellite images. And those sulfide eruptions can cause things like fish die-offs and so on. But those conditions are the conditions that are that enrich for these giant sulfur oxidizing bacteria. And these bacteria are chemolithotrophs primarily, although they're metabolically diverse. They can also use organics, but typically they're using sulfide or reduced sulfur compounds as an electron donor and oxygen or nitrate as their electron acceptor for their metabolism. And because they live in these conditions that are kind of variable with respect to the availability of electron donors or acceptors, these organisms are really built for storage. So they have a capacity to store nitrate within the cells, as well as sulfur, as well as carbon, and also importantly, phosphorus in the form of polyphosphate granules, which was originally shown by Heide Schultz and Schultz in kind of groundbreaking work in 2006. And I first got interested in these bacteria because of their kind of association with phosphorites. And phosphorites are basically sedimentary rocks that are basically derived from the reworking of marine sediments that are rich in calcium phosphate. And so you can have basically enrichment, you can have the precipitation of carbonate, fluorapatite, and other similar phases that then gets basically kind of worked through, reworked or concentrated through sedimentary reworking to make these kind of accumulations of calcium phosphate minerals. And these phosphorites are kind of relevant and important to us, to humans, because basically we increasingly rely on mining these mineral deposits as a source of phosphorus for agriculture. And I should say that the phosphorite mines are primarily ancient deposits that are now ancient marine deposits that are now on the continents. And so those are the principal source of mining of phosphorus is on land, right? But more recently, there has been an effort by some mining companies to get access to mining phosphorites on the shelf of Namibia, where you have these kind of extensive, very recent and currently forming phosphorites. And so this is kind of a new and rather controversial effort to start mining the seafloor for phosphorite rather than just mining it on land. And there's kind of ongoing debate about that in Namibia. So there was originally some permits that were given, but then those were kind of put on hold while there was kind of more study going on. And currently, I don't think that they've actually started doing anything. I don't know what the exact status is, but I think the last time I checked, it was kind of on hold. But that's kind of an interesting, and I guess depending on your perspective, I guess from mine, it's a little bit disturbing prospect of actually now going after phosphorus on the ocean floor, particularly in upwelling zone where you have major kind of fisheries that act as kind of food for a lot of people in that area. But again, I guess it's worth mentioning that phosphorus is not really or at least these phosphorite deposits are not really a renewable resource, and we're kind of mining our way through them. And so that's something that we're gonna, phosphorus availability is something that we're gonna, likely confront in our lifetime. Phosphorus availability problems. So if we look at where phosphorites are forming today, most of them are principally, you can see in these red dots here, principally they're forming in upwelling zones, like you can see off of Namibia here and off of Chile. And you also have, these are also sites where you have relatively recent or relic phosphorites at the seafloor. And these roughly can correlate with, interestingly, with environments where we see these giant sulfur bacteria as well. And so the kind of correlation between, there's kind of a spatial correlation between some of the phosphorite deposits on the Namibian shelf. So you can see here, this is on the lower left, this is off of Walvis Bay, and the red areas kind of highlight where there's concentrations of phosphorite. And then up on the top panel here, this is kind of showing how organic rich the sediments are here. So this is like 10 weight percent TOC. So this is like really, really organic rich, extremely organic rich. It's like If you look at these sediments, when you collect them with a core, it looks like a kind of like a diatomaceous ooze. But then there are pockets of phosphorite kind of interspersed throughout here. And so one of the, in some cases, there's basically you find these areas that are enriched in Thiomargarita and other giant sulfur bacteria that are closely associated with the phosphorite. And this kind of led to the theory that Schultz and Schultz developed, and over a number of other people have worked on it over the years, of this kind of association between Thiomargarita and phosphorites as basically being driven by a mechanism that involves polyphosphate, whereby kind of under oxic conditions, Thiomargarita takes up phosphorus and stores it as polyphosphate. And then under kind of anoxic conditions, polyphosphate is broken down and then releases that phosphate to the surroundings. And so basically the idea is that Thiomargarita could act as kind of a lens for focusing phosphorus from decaying phytodetritus into pore waters where calcium phosphate phases can precipitate. And Brock and Schultz further showed that the mechanism, or at least the trigger for the phosphate release, was not just anoxia, but was more specifically sulfide. At least these results suggest that. And so the idea here is basically that in Namibian shelf sediments, where you have lots of Thiomargarita cells, you have this kind of correlation with peak in pore water phosphate, which can get up to several hundred, almost millimolar. I've actually seen concentrations in some of these sediments. I should say several hundred micromolar or almost millimolar concentrations. And then you can see that this pore water phosphate peak correlates roughly with mineral phosphate as well. So this is where you have phosphorite as well. So all these things kind of co-occur, and Schultz and Schultz proposed that the mechanism for this was that the linkage of polyphosphate concentrating phosphorus within these kind of pore water zones. And there are other environments as well where we can see an association between phosphorites and sulfur bacteria. These are upwelling sediments off of Costa Rica, and this is the view out the front of the submersible Alvin. And so these are very extensive mats of sulfur oxidizing bacteria, including Beggiatoa. So here are some Beggiatoa mats, and basically you see here some filaments. We collected these filaments from these Beggiatoa mats that have both elemental sulfur in them that show that they are Beggiatoa filaments, but then also they're partially mineralized with apatite. So there actually is carbonate fluorapatite that's precipitating on these filaments. So we can see that there's other environments where we see a close kind of association between phosphorite formation and sulfur bacteria. On the other hand, there are other environments, though, that seem to not kind of show this clear linkage. For example, off of Chile, some researchers have shown that you have Thioploca, extensive kind of Thioploca mats there, and you have a pore water phosphate peaks, as well as mineral phosphorite in the sediments there. But really, apparently, the Thioploca don't show any evidence for polyphosphate accumulation, or at least at the time of the study, they didn't. So that's kind of interesting. And also in our some of our own work, we took some phosphorite grains off of Namibia, and we cleaned them, and we basically extracted DNA from them after we cleaned them and crushed them up. And we basically kind of looked at the communities that were preserved as DNA within the phosphorite, and we can compare those with basically the sediments, the communities that were in the sediments, the surrounding sediments. And we see that there's actually enrichment in the phosphorites for a number of different clades of organisms, including the representatives of the Disulfobulbaceae. You can see it here. These like kind of the red peak is kind of showing the OTUs enrichment over like the symbols here, which are the different kind of OTU counts in the sediment surrounding sediment. So this is the phosphorite in the red line versus the surrounding kind of background sediments. Phosphorite DNA hosted by the phosphorite. And so there are some organisms here, or some DNA, preserved DNA in the phosphorite that represents organisms that do indeed have the capability for making polyphosphate. So if we look at their genomes, they have, genes like PPK and PPX. And so potentially, and some of these organisms like sediment cola, we know for sure that they can store polyphosphate as well. So these are potentially other polyphosphate accumulating bacteria that are living in these sediments as well. Granted, Thiomargarita is very large cells, and so potentially they have more kind of capacity for phosphorus focusing, but then also potentially these little bacteria are also extremely numerous. Right? So does polyphosphate metabolism by giant bacteria kind of control the phosphorite formation off of Namibia? I think there's some pretty strong evidence, including radio tracer work and so on, that I didn't get into, that suggests that they are quite important. But there's also some observations that in other places that may not quite fit with that, and it may be that there's kind of more complexity to the story. And so I think that kind of more additional work on this needs to be done. But I think it's a really important potential mechanism, and I think it's really something that needs to be further kind of explored. And we could also ask the question of whether or not these polyphosphate accumulators are represented in the fossil record as well. A number of ancient phosphorites actually contain microfossils that could be interpreted as polyphosphate accumulating bacteria. For example, these filamentous structures here in Neoproterozoic rocks from China, they have what are basically kind of sulfur inclusions in them, or sulfur that's associated with organic matter, as opposed to sulfur that's associated with pyrite. There's also some of that too. You can see here in the element maps. But there's some sulfur here that is just associated with organic matter in these little kind of spots. And these things, in terms of their morphology, they could be interpreted as cyanobacteria, or they could be interpreted as Veggiatoa-like bacteria, right? But the preservation of this sulfur is suggestive that they may be, in fact, Veggiatoa. And so perhaps those phosphorites represent kind of a similar habitat and depositional environment as you see as off of Namibia and off of these kind of upwelling zones. So that's a possibility. And then we can even go back to older in the Proterozoic, Mesoproterozoic phosphorites from India. And these are basically some of the oldest phosphorites in the form of phosphatized stromatolites. And these, my PhD student, Chris Crosby, found these structures in there that are that have these kind of twisted stalks of iron oxides that are very reminiscent of filamentous iron oxidizing bacteria. These also contain organic carbon that's of age, or a thermal characteristic that indicates that it's syngenetic organic carbon. And so these in some cases, modern iron oxidizing bacteria are known to accumulate polyphosphate as well. And at least these perhaps may serve as an indicator that this is in an environment, this depositional environment was conducive to kind of the conditions that would enrich for polyphosphate accumulating bacteria. Okay, so now that's kind of the first part of what I wanted to talk about. Now I wanted to turn to a different kind of environmental condition, and that's polyphosphate in the human oral cavity. And so I think working on polyphosphate metabolism in the ocean environment got myself and collaborators that thinking about this idea of what other kind of environments could this polyphosphate metabolism be important in? And so one of those environments is the human oral environment. And there, kind of the canonical view is that you have dental caries or cavity formation that results from basically the metabolism of sugar by microbes in saliva and plaque that then can basically cause dissolution or demineralization of the calcium phosphate minerals that make up our teeth, right? Hydroxyapatite and so on, just like they're basically like rocks that are covered with biofilms, right? But this view also doesn't really kind of look at the kind of geochemistry of the and the saturation state of the environment, the microenvironments that are in equilibrium with the tooth surface. And so we could actually think about dental caries as the result of undersaturation with respect to calcium phosphate minerals like hydroxyapatite, whereas we could also think of dental calculus as kind of a supersaturated regime in which the precipitation of calcium phosphate minerals is favored. And so this kind of led to this hypothesis that maybe polyphosphate accumulating bacteria in dental plaque could play a role in either dental caries or dental calculus, depending on the kind of the geochemical microenvironment and the conditions of the cells at the time, wherein you basically have cells in the plaque and saliva that could be taking up phosphate and also potentially calcium as a counterion and storing those in the cell, and then creating undersaturated conditions locally that could lead to tooth dissolution. Alternatively, in other environments, like along the gum line, you could have the release of this phosphate or calcium that basically kind of leads to supersaturated conditions, which would cause, for example, dental calculus to form. And so, is polyphosphate metabolism actually something that occurs in oral taxa? Well, we can kind of look at this in a number of different potential ways. There are different genes that are known for polyphosphate metabolism, like polyphosphate kinase 1, PPK1, which is involved in the synthesis of polyphosphate, or other genes which are associated with or used for, I should say, that code for the enzymes that are responsible for hydrolyzing the polyphosphate, things like PPK2 or exopolyphosphatase. And so we can, Dr. Beverly Flood looked at some of the genomes of different oral taxa, and you can see here, these are different groups like Neisseria, Rothia, Cyanobacteria, these different groups that are known organisms in the oral environment. And a number of them actually have polyphosphate genes that are present and known in their genomes, and also in many of these taxa, there are actually decades of literature that show that they actually do accumulate polyphosphate. And some of these organisms are associated with healthy kind of oral flora or conditions of a microbiome that's associated with kind of healthy individuals, versus in other cases, some of these groups are associated with caries prone individuals. And so in some cases, some of these groups are associated with both. So it can be quite complex. There are other organisms out there that don't appear to have at least the kind of canonical genes for polyphosphate accumulation, like strep mutans, which is kind of a famous caries associated taxon. So it does seem that there are lots of these kind of oral taxa do have the potential to the genetic potential to at least make polyphosphate, but purely bioinformatic approach can be kind of challenging, in that there aren't genes that are so far recognized as being responsible for the actual accumulation of polyphosphate versus just its synthesis. So it's difficult to tell if an organism is actually a polyphosphate accumulator versus just having a genetic potential to make polyphosphate or hydrolyze it. But my former master's student, Ash Breland, did some work showing that there are, in fact, if we DAPI stain plaque and saliva, it's full of polyphosphate accumulating bacteria. Here you can see all sorts of different morphotypes and of different organisms in dental plaque that have, you can see here the yellow, the bright yellow, or the kind of green, depending on the microscope conditions. This is all inclusions of polyphosphate. In some areas of the dental plaque, it can be quite heterogeneous. So there's, in some areas there might be not very much, in other areas there can be pockets where they're extremely dense polyphosphate accumulators, and the cell concentrations basically are basically equivalent to kind of the culture conditions that we worked with in model environment organisms like Lactobacillus rhamnosus, where we basically can grow this organism, which is a known kind of oral taxon, and we can look at the kind of the phosphate drawdown that occurs under conditions in which Lactobacillus makes polyphosphate versus conditions where it doesn't. So if you limit manganese in the case of Lactobacillus, they basically don't make very much. They make very little polyphosphate versus conditions where they have it. They make quite a lot of polyphosphate, and so we can kind of compare the conditions where they're making polyphosphate with ones that they don't, and kind of quantify and get an idea of the amount of phosphate that's being taken up specifically by polyphosphate, and not just like general cell growth, right? And then we can take that and we can kind of apply that to looking at geochemical modeling and kind of look at the potential influence that this kind of phosphate drawdown could have on saturation state. And then indeed, there's quite a lot of variability in terms of human saliva, the concentrations of phosphate and calcium, as well as lots of heterogeneity in terms of pH, which is also influenced by microbes and diet and so on. So it's quite complex. But there are some kind of conditions, intermediate conditions, where you have basically conditions like this, where you have a pH of 5.4, and here are the calcium concentrations, and here's phosphate concentrations. And basically you can have phosphate concentrations that are starting kind of allowing the saliva to be kind of near equilibrium or near saturation. And then basically the uptake of the phosphate by polyphosphate accumulating bacteria could draw the phosphate down to kind of undersaturated conditions. Now, there are other concentrations of phosphate and calcium that are found in other individuals where maybe this drawdown actually wouldn't influence them one way or another, but this is kind of showing some of the range of like the different phosphate and saturation states that are actually possible in the mouth. And there's quite a variable, and I'm not showing all of it, but there are conditions where you can get this kind of crossing of saturation from supersaturated to undersaturated. And really, well, then under other conditions, you could potentially have that phosphate released by the polyphosphate accumulating bacteria, and then it could go back the other way. And this could could influence this kind of canonical model of demineralization and remineralization that's long been kind of known in the dental oral health world, wherein a typical individual undergoes kind of mineralization and demineralization over time. And so in the case of people that are having caries progression or cavity progression, you have basically more kind of demineralization than you have remineralization. And so this cycle is kind of going down, and that's what's causing caries progression. Versus kind of the other side of it is the kind of reversal of caries, where you have kind of more remineralization and less demineralization. But there still is these kind of cycles of remineralization and demineralization, which are kind of typical of what happens in the oral environment, broadly speaking. And for most of us, I think that's because our saliva is kind of perched near saturation, and so there are swings that go up and down, and those things can be influenced by our diet and so on. But what we're suggesting is that polyphosphate accumulators could play a role in that kind of modulation of going up and down across something that's close to saturation. And again, if we look at, you might say, well, Thiomargarita was influenced by sulfide. Maybe that's something totally different in the mouth. Indeed, a lot of these oral taxa, their usage of polyphosphate is much different than like kind of the canonical gram negatives, and they can use polyphosphate for many different things, oftentimes in different ways than you'd find in gram negatives. And certainly, things like they can, as I said before, phosphorylate glucose. That's something, a different use of polyphosphate than say maybe energy storage response in response to sulfitic conditions, right? So basically that we have to understand kind of what the conditions are like that influence these polyphosphate accumulating bacteria in the mouth, if we want to understand how like oral conditions could actually influence polyphosphate phosphorus uptake and release. And so this is kind of work that's in progress by our postdoc Subhangshu Mandal, and I'm not going to show any of his data yet because he still got it in progress, but that will hopefully be upcoming in the near future. But he's basically kind of looking at the different environmental factors that kind of influence phosphorus uptake and release. And then another kind of open question is, who are these major polyphosphate accumulators in the oral environment? You can see them with microscopy, but we don't really know who they are. And so one of the things that we're doing right now is we're trying to use flow cytometry basically to kind of separate out and cell sorting to separate out the cells that do have that accumulate polyphosphate, that we can basically kind of label or stain with DAPI and separate those from those that don't. And so that work is kind of also ongoing. It's been kind of a challenging effort, but we basically want to kind of calibrate the separation using cultures of pure cultures under conditions where they make polyphosphate versus conditions where they don't. You can see there's kind of a nice separation here. And so we're basically working with pure cultures until we can be kind of confident in our ability to separate cells of different sizes and so on. But then what we're going to do is basically isolate these polyphosphate accumulating bacteria and then do amplicon or other kind of sequencing and bioinformatics approaches to kind of characterize the populations that are accumulating polyphosphate in the mouth. Okay, so that's kind of work that we have ongoing, and I'd like to thank our sponsors, NSF, Simons Foundation, and National Institutes of Health. And I want to thank all of you for having me here, and I'm happy to take your questions.
Thank you very much for this really interesting talk. So if you have any question, you can either raise your hand and we will give you the thank you, Karim. You can also write it in the chat. I maybe just have a quick question before. It's really a naive question, but you are talking about polyphosphate-accumulating bacteria and sometimes about super accumulator, but where is the threshold in that? How do you define?
Yeah, I guess that's a good question. I mean, you could extract the kind of polyphosphate and kind of quantify it, but really most of the more or less most of the bacteria that we think we associate with storing a lot of polyphosphate, they store it as inclusions in the cell, and so those are inclusions that you could image with DAPI staining, which isn't that great. We're actually working on alternatives to DAPI staining because there can be kind of non-specific binding and things like that are a concern. So we're actually trying to develop some alternative probes to DAPI, but also, I mean, you could do it with TEM or other ways, but you can actually see like a granule, and these granules have been studied for a long time. They were recognized more than 100 years ago, I think, but that's typically how we think of it, although polyphosphate could be accumulated in cell in other ways as well. So that's a good question, and it's sort of a little bit subjective. But
okay, so it's the presence of these granules more than the number of these granules, if I understand.
More or less. Well, I think granules at all, yeah.
Okay.
I mean, granules at all, yeah. And then some of them could be bigger or smaller, or more numerous. Yeah.
Okay, thanks. Karim, if you want to ask your question.
So thank you very much, Jake. I was struck by one of the, I mean, I've seen that several times before, but I mean, when you were reviewing the work on Thiomargarita, I realized at some point you had some depth profiles of the number of cells and the chemical composition of the solutions, and you see that basically there is some correlation between the orthophosphate concentration in the solution and the number of cells.
Right.
So that basically tells us that those cells accumulate phosphorus, but they also release some, and this is why you get both higher amount of phosphorus in the cells and also higher amount of phosphorus in the solution. I was just, the first question was, I was wondering if this kind of correlation has been seen somewhere else by some other people. Do you know that? I mean,
it's a good question. We see off of Namibia, we see areas where you have Thiomargarita that have polyphosphate in it, and there's no phosphorite in that area. And you also see lots of phosphorites where there's no Thiomargarita as well. But then it's complicated because the idea is that these kind of localized supersaturation conditions can cause like the authigenic precipitation of apatite, but then it takes like the sedimentary reworking to actually kind of accumulate like an actual mass of phosphorite. So that having phosphorite without Thiomargarita, well, those could be thousands of years old, and now that those zones have migrated or something like that. But it is true that it does seem like that there's not always this like kind of clear correlation between where we have Thiomargarita and we have pore water phosphate enrichment. And then, in fact, that's what I was trying to show with that Thioploca data.
Okay, and then a second question. So on your second part, I mean, this is pretty cool because I mean, it's been a few years we have started working with medical scientists, but not on teeth, but on urine, where the formation of kidney stones, and basically we also try to follow the track of this similar stuff, like could we have polyphosphate stuff in some infections. One question I would have is that, so basically, I guess one of the question is whether, when do you uptake or when do you release phosphorus by those polyphosphate accumulating bacteria compared to the phosphorus level where basically you have saturation with hydroxyapatite. Of course, it also depends on calcium concentration and pH, but let's say if, I don't know, some micromolar of phosphorus you're at saturation, I guess the question would be that, are your bacteria accumulating or releasing a phosphorus? And so I guess the project of your students probably to look also for other parameters impacting that, right?
Yeah, exactly. That's what we're trying to do. And like correlating kind of, growth stage and different environmental conditions, different substrates, like different, different carbon sources, different pH conditions, a whole bunch of different parameters basically is what we're looking at to see which ones are important for controlling the polyphosphate accumulation. And then mostly we've been focused on that right now, but then the release is also, a part of that too. And it's an interesting question too about the release. Like it's definitely seen in some organisms, this release of phosphate, but it isn't clear to me like that you always have to have that release. Like you could just, make it and hydrolyze it and then remake it or whatever without dumping it into the environment, I would think. Or at least go into the periplasm if you're, importing protons or something versus just completely releasing it. So that's another, kind of aspect of this that I think is, we have to figure out. But yeah, there's a lot to look at in terms of the different environmental conditions and the timing and so on. So that's what we're trying to do. And these gram positives, they seem very different from, the gram negatives. And so working with them is interesting and challenging. And it's also hard to do things like, a lot of the kind of fish methods and things like that are challenging with gram positives because they have thick peptidoglycan and so on. So a lot of the things that you think would be kind of simple have been like taking us a lot of time. And so, yeah. Yeah.
I bet. Thanks.
Thanks for your questions.
So we have another question from Francois.
Yeah, thank you, Jake. I was just wondering, do you see any systematics with cations that are related in the polyphosphates with the conditions of accumulation of the polyphosphates, both actually in the phosphorites or in the phosphorite conditions, or even in the oral cavity, or what are there always calcium polyphosphates, or is there systematics on that? And can you tell us something about the mechanism itself?
It seems to depend on the depend on both the organism and the environmental conditions and what kind of cations are available. But some organisms, like for example, lactobacillus, they preferentially take up manganese and they store that as a cation, and they use that manganese actually as an antioxidant, really. So they have that, it serves as an antioxidant role. And interestingly, if you just don't give them any manganese at all, then they don't make very much polyphosphate, which is interesting.
Yeah.
But I think you can give them a bunch of magnesium instead, and they'll use that, or calcium, right? And so they could take that up and concentrate it as well. In some cases, it seems like there isn't really like, as far as I know, that there isn't like a clear cation in some cases, but in other cases, it's a very strong accumulation of this cation within the granule. And it seems in some organisms, it's used as like a scavenging of toxins potentially, so they can store toxins in the cell as well. But yeah, I think it depends on the organism and if they get any kind of benefit out of it, and also yeah, I mean what they're basically kind of what they have available to them. So what it's both genes and environment, I would say. And so it's different from organism to organism.
And actually a related question is about the phosphorus to cation ratio and things like that. Are there hints about the chain length in the polyphosphate as a function of conditions, and maybe about even the hydration rate of the polyphosphates? I mean, it's because it will affect a lot the polyphosphate itself, right? So are there studies about that and chain lengths and things like this?
I think there's some, but I actually I could take a look and try to send you something if I can find. I've seen some stuff, but I don't know. I think there's some literature out there, but probably not a ton of it.
Okay.
Yeah, and some of it I think is older literature. So
okay.
Yeah.
Thank you very much.
Thank you. If you want to put your email in the chat, I could try if I run across something.
Yeah, thank you very much. I will.
Okay.
Elodie, if you want to unmute your mic.
Yes, thank you, Jake. I have one question on the cytometry experiments. I'm not sure to really understand. Is it one pure culture in which you have both cells with or without polyphosphates, or did you combine or pool several cultures that accumulate or not polyphosphates?
We've done both. So we started off by just taking like pure culture that we know has polyphosphate accumulators in it, and then we kind of look at that, what that gate or window, that cytometry data looks like, and then we did them separately with we use the same organisms that didn't have polyphosphate in them, and we see where they plot, and then we mix them together, and then we sort them. And what we're doing right now is we're seeing if we can, get under the microscope, we can see that we sorted the right stuff. And then also the challenge in the plaque is that you also have some bacteria that are smaller and bigger. Like you have like these big cyanobacter, and you have really small stuff like Neisseria. And so we want to make sure that we can capture those as well, or at least know what we are capturing and or not capturing. And so that's what we're doing now, is we're using other pure cultures of bacteria of other sizes and other that also have different polyphosphate kind of characteristics as well, so that we kind of have a characterization of the different cytometry characteristics, and so we can have a sense of what we want to separate out versus what we don't. And then again, separating those and looking at them under the microscope to see, are we getting what we think we're getting? So that's where we're at. Thank you.
François, is your hand still raised from last time, or do you have another question?
No, sorry. I forgot to remove it. Sorry.
Okay, no problem. Do we have another question in the chat, maybe? I don't think so. So I think we are right on time to end this seminar. Okay. So thank you again, Jake, to present your work, and it was really, really interesting. And thank you all for coming and joining us, and see you in 2 weeks for another seminar.
Great, thank you so much for having me, and it was nice meeting many of you and seeing a few familiar faces. It was great to see you, and hopefully I can see you all again in person sometime soon.
Yeah,
that's so
Take care all.
Thank you very much. Bye, Jake.
Bye.
Bye. Thank you.